This is Unit 9, Mendelian Genetics, looking at 9.1, Meiosis. That's how I pronounce it. Some people say Meiosis. I say Meiosis. Doesn't matter. Let's talk about it. Meiosis is, we're not talking about meiosis yet. We're going to talk about chromosomes, because that's pretty important. You need to know about chromosomes before you get to meiosis. So the average cell in an organism's body, sexually reproducing body, is going to be a diploid cell, meaning that the chromosomes in that cell are going to represent one set of chromosomes from mom, the other set of chromosomes from dad, right? And so you have those chromosomes exist in pairs, and we're going to call those pairs a name. They're called homologous pairs. Homologous doesn't mean identical. They're not identical, right? Mom's chromosomes and dad's chromosomes aren't the same, but they do have the same parts on them, right? They have the same they have the same genes on them. And so chromosome 1 from mom is going to be similar to chromosome 1 from dad. They're not going to be identical because your mom and dad don't have identical genes, but they're going to be homologous. That'll make sense as we go through this rest of this unit. All right, and then that individual can then make haploid cells, which are sex cells. We'll get to that in a second. And those haploid cells have half the number of chromosomes, just 23, if you think about it, right? 23 plus 23 is 46. Why am I using these numbers? Well, in humans have diploid 46, haploid 23. It's not the same for dogs or giraffes or other organisms, right? So I'm just going to use those numbers as an example, but it could be anything. And so for humans, for instance, 2n, which is what diploid is, would be 46. And so 2n equals 46, then n equals 23. N is the prefix or the symbol for haploid. 2n is going to be the symbol for diploid. That idea of homologous pairs, very important. You see how they're represented here, kind of the pink-purple thing. They're not identical, but they're the same size. They have the same genes, blue, light blue, green, light green. You get the idea. Homologous pairs, very important concept. Moving on. What is meiosis? It's the process of making gametes, haploid cells, in eukaryotic organisms, specifically sexually reproducing organisms. Meiosis exists in two processes, Meiosis I, Meiosis II. It basically represents two divisions because the product of meiosis is 4 genetically different haploid cells. You take one thing, you divide it, you have 2 things. You take those 2 things, you divide it, you have 4 things. That's kind of the idea, right? And so that diploid cell that you start with is going to replicate. The DNA is going to replicate. That happens during interphase. And so then you have twice as the number of DNA, and then that's going to divide once, and then it's going to divide again. And so you go from 2 to 1 to halves, right, if you want to think of it that way. In human cells, we have 46 chromosomes. After interphase, it's 92 chromosomes. During meiosis I, divides, and you have 46. And during two cells with 46, and then in meiosis II, you have 4 cells with 23. Just to help you with the math, right? It could be any organism, right? But we're going to use humans because, well, we know the context. So let's talk about meiosis I. Interphase 1, or there's no interphase 1, there's just interphase. It's already happened. DNA is replicated in interphase. It's important. And then you see some familiar friends here. Prophase 1, metaphase 1, anaphase 1, telophase 1, right? These are the names of the phases. Very similar to mitosis. Not the same, but similar, right? We're going to kind of hit the high points. I'm going to mainly talk about prophase 1 and anaphase 1. Not to say those other things aren't important, right? They're important, but we're going to mainly hit the high points here. So let's talk about prophase 1. During prophase 1, the nucleus is going to disappear, all the prophase things that you remember and you love. But the important thing for meiosis I, for prophase 1, is that crossing over occurs. Tetrads form and crossing over occurs. What is a tetrad? Well, the word tetra, the prefix tetra means 4, right? And so a tetrad is when you have 4 pieces. Notice there you have 4 pieces. You have a homologous pair, right? And each pair has 2, those sister chromatids. Remember that from a previous unit. So the pink chromosome is a duplicated chromosome, so it has 2 sister chromatids. They're identical to one another. The blue chromosome represents the homologous pair of the pink chromosome. It has 2 sister chromatids. They're also identical to one another. This homologous pair is going to kind of line up with each other, right? And those homologous sections are going to cross over, which is exactly what you see happening here. It'd be similar to you taking like the end of your index finger off of one hand and putting it on the other one. Essentially the same function. It's going to look different, right? but it's essentially the same function. It's a homolog, if you want to think about it that way. This happens during crossing over. It's going to create lots of variation. That's all you have to know about it for here. Go back to Meiosis I real quick, because we're going to talk about Interphase I. During Interphase I, something very important happens. Homologous pairs are going to separate. So if you see this on this picture, you can see I like this picture because it shows that crossing over has occurred as well. But this is where red separates from blue in this particular picture. If you want to go back to this one, it's also red and blue. This one's pink and blue. Maybe red and blues are a good color. I don't know. So this is where the one that dad gave you is going to separate from the one that mom gave you, right? Homologous pairs are separating, and then telophase is going to happen, cytokinesis. You're going to have two cells, and those two cells are haploid, right? Even though they have twice as much chromosomes as you'd expect, they're haploid because they don't have a homologous pair with them, right? And so now you have two cells after Meiosis I, and that next moving on to Meiosis II. All right, that's not Meiosis II. This is Meiosis II. Very similar. Prophase II, Metaphase II, Anaphase II, Telophase II, cytokinesis, all the same stuff. However, we're going to hit the high points, and that high point is Anaphase II. And during Anaphase II, what's happening? Sister chromatids are going to separate. Right, and so homologous pairs separated in Interphase I, sister chromatids are going to separate in Interphase II, and so that after telophase and cytokinesis, you're going to have four cells, each one, because of crossing over, because of those homologous pairs going to be separating in different ways. We'll get to that later. You're going to have four genetically different cells. Pretty important. All right, let's talk about gamete formation. It's going to be different for males and females. So for males, production of sperm, you don't need to know the exact name of it that you see there at the top. It's a good name, but you don't need to know it. You start with the one diploid cell, has a name, don't need to know that. That cell is going to divide once. You're going to have two. Those cells are going to divide again. You're going to have four. Each one of those sperm cells is genetically different. That's it. Sperm production is pretty boring. Egg production, it's a little bit different. It's a bit different. All right, and so you have this 2N cell at the very beginning. That cell is going to divide. You have two cells there, right? And then those two cells are also going to divide, but notice one of the cells is much bigger than the other three. All right, those other three cells are going to be called polar bodies. Polar bodies are not going to be used by the organism. Some organisms use them for other things, but we're not going to worry about that. There's going to be one egg that's going to be produced from this process that's usable. Egg cells have to contain all the cytoplasm of the first several divisions, and so egg cells are typically much bigger than the rest of the cells. And so you can imagine these little polar bodies are just tiny little things. They've got nothing in them. They're going to be kind of reabsorbed by the body, whereas the eggs are going to be, that's what's going to be used. So during egg production, only one egg is made. It's a haploid. During sperm production, 4 sperm cells are going to be made. All right, so let's talk a little bit about nondisjunction. Nondisjunction is when chromosomes fail to separate. Okay, when chromosomes fail to separate during Anaphase I or Anaphase II, this causes nondisjunction. What is that going to result in? You're going to have cells, you're going to have gametes that either have extra chromosomes or have fewer chromosomes than they should. You see that in the examples there. You have some cells that have it's almost like an N+1. It is that. You don't need to know that, but that's what it is. Some are N-1. They have one less than you'd expect them to have, right? It's just going to cause problems. So that sperm cell or that egg cell fertilizes a normal. One of those pairs is going to have 3, right? There's some things that are associated with this. One of the most common that you've probably heard of is called trisomy 21, or we call individuals that have trisomy 21. They have Down syndrome because it is every one of their cells, the 21st pair has 3 trisomies, where you get that from. And so this can cause systemic kinds of issues. So nondisjunction, think about that word. It's a failure to separate during Anaphase I or Anaphase II. One more thing. Make sure that we understand the difference between mitosis and meiosis. In meiosis, you have one division. Both cells, or you have a division, and the result is 2 identical cells, both diploid daughter cells, and they're identical to one another. At the end of meiosis, which is specifically for the production of gametes, you have 4 haploid cells that are genetically different from one another.